Good morning to all my dear students. Welcome to our Zoology online class. So in the last week, we have written the half-yearly examination. I really appreciate you all. You did the exam very nicely, and you have written and you have scored good mark. So I really appreciate you all. Okay, my dear students. Today we are going to start a new topic. Okay, our topic name is immunology. Okay, our topic name is immunology. See, immunology means studies about our immune system. You all know our body is made up of many systems: digestive system, respiratory system, nervous system, skeletal system, like that many systems. So our body is made up of different systems. So here immune system is one of the very important system, and the main function of this immune system is protecting our body from the diseases. Okay, so the main function, every system is performing some important functions in our body. Okay, like that here this immune system performing one important function. The name of the function is protection. Okay, protecting our body from the diseases. Okay, so studies about our immune system is called immunology. Understand? Then two more question: immunity. Immunity meaning the overall ability of our body to fight against the disease causing microorganism to protect our body. So the overall capacity or overall ability of the body to fight against the pathogenic microorganism is called immunity. Okay, it's called immunity. Understand? And our immune system has some different characters. Okay, some specialized character. For example, say our immune system can destroy and eliminate the foreign particles which are entered inside the body. Here these foreign particles may be microorganisms or some toxins or chemicals or any types of foreign particles. Okay, so when dangerous foreign particles enter inside the body, immediately our immune system only destroying that foreign particles and eliminating it from the body. So this is a special character of our immune system. Okay, then second one is self and non-self. Okay, distinguishing the self and non-self. That means our immune system can realize which molecule is self. That means which molecule is our body molecules and which molecule is the foreign molecules. Okay, so our immune system can recognize the our molecules and can recognize the foreign molecules. Okay, so this is the next specialist character of our immune system. Okay, and sometimes our immune system fail to recognize the foreign particles and self particles. Sometimes our immune system destroying our own cells, our own self cells. Finally, it leads to some diseases. These diseases are called autoimmune diseases. These diseases are called autoimmune diseases. Understand a few? Okay. Now we are going to study about two types of immunity. Okay, immunity meaning the overall ability or capacity of the body to fight against the pathogenic microorganism is called immunity. And the immunity is classified into two major categories. Okay, the first major category is innate immunity. Innate immunity, and second one is acquired immunity. So these are the two major categories of the immunity: innate immunity and acquired immunity. Here, innate immunity means the natural immunity, otherwise called a non-specific immunity. Okay, so the human beings and all the every living organisms getting some immunity power or immunity right from the birth. Okay, so naturally some immune power is present in our body, so that immune power is called innate immunity. Okay, so all the organisms getting this immunity, this innate immunity right from the birth, Understand a few? Then next one is, say, this innate immunity protecting our body by using some barriers. Here barriers means that I had. So by using some barriers, this innate immunity protecting our body. Okay, so the first barrier is anatomical barrier. First barrier is anatomical. Okay, so first barrier is called anatomical barrier. Example for anatomical barriers are skin and mucus. Okay, so skin and mucous membrane are the example for the anatomical barrier. Here, the pH of the skin is very low. pH of the skin is very low. So, skin prevent the entry of microorganism inside the body. Prevent the entry of microorganism inside the body. Then the second example for this anatomical barrier is mucous membrane. You all know that this mucous membrane is present in the inner side or the inner lining of the digestive system, respiratory system and urogenital system. So whenever a microorganism enter inside the body, immediately these microorganisms attach or trap that microorganism. Finally, that microorganisms are destroyed. Okay, so by using these two barriers, our immune system protecting our body. So skin and mucous membrane, these two are the example for the anatomical barrier. Okay, the second barrier is physiological barrier. Okay, physiological barrier. Example for physiological barrier is very best example is temperature. Okay, our body temperature will not allow the normal microorganism to grow. Okay, particularly during fever time. Okay, during fever the temperature of the body is very high. So this high temperature prevent the growth of other microorganisms. So, body temperature is one of the example for the physiological barrier. Body temperature is one of the example for the physiological barrier. Understand a few? Then, see, next one is a low pH. That means the hydrochloric acid, you all know that acid. Hydrochloric acid is highly secreted inside the digestive system. So, when a microorganism is entered inside the digestive system along with the food or water, immediately the hydrochloric acid which are present inside the digestive system is destroying that microorganism or killing that microorganism. Okay, so temperature, low pH. Then the third example for this physiological barrier is some chemical mediators. Chemical mediators means lysozyme. Lysozyme is a enzyme present in the saliva and also in the tears. So whenever a microorganism bacteria is entering inside the mouth or eyes, the lysozymes which are present in the saliva and tears destroying or break down the cell wall of the bacteria. Okay, and the second example for this chemical mediator is interferons. When our cells are infected, particularly the WBCs, okay, when our WBCs are infected with the virus, immediately they are releasing a chemical like substance which is called interferons. These interferons protecting the other cells from the viral infection. Okay, so for the chemical mediator, now I said two important examples. One is the lysosome and the second one is the interferons. Okay, so temperature, then hydrochloric acid, then chemical mediator. These three are the example for the physiological barrier. Okay, then next one is phagocytic barrier. Okay, phagocytic barrier, some specialized cells. Okay, for example, monocytes, the neutrophils, macrophages. Okay, so these cells are otherwise called phagocytic cells. So these phagocytic cells engulf the microorganism which are entered inside the body. So when a microorganism or a foreign particle is entered inside the body, these phagocytic cells engulf that microorganisms. Thus, they are protecting our body from the diseases. Listen a few. Then, the last barrier is inflammatory barrier. Inflammatory barrier. See, inflammatory barriers means when our tissues getting damaged or if any injuries occur in our body, in that damaged area or injured area, some vascular fluids are released. Okay, so these vascular fluid consist of some chemical like substance. Okay, histamine, serotonin and prostaglandins. So these three chemical like substances are highly present in the vascular fluid which are released in the damaged areas. Okay, so these chemical like substance gather all the phagocytic cells in the affected area. Thus, it prevents the entry of microorganism in the affected area. Okay, understand you? So here inflammatory barrier meaning So when a tissue is damaged or if any injuries occur in our body, the damaged tissue releasing a vascular fluid. That vascular fluid consists of three chemical-like substances, okay, histamine, serotonin and prostaglandins. So these chemical-like substances gather all the phagocytic cells in the affected area. Thus they are protecting our body from the diseases. Okay, so this phenomenon is called diabetes. Okay, diabetes. Okay, so by using all these four barriers, our body protecting our body, our immune system protecting our body from the diseases. Understand a few? So now we have completed the innate immunity. Okay, the second one is acquired immunity. Second immunity is called acquired immunity. Acquired immunity means the immunity which is produced inside the body after birth. Okay, so after the birth, that means during our lifetime we are getting some immunity power. That immunity powers are togetherly called acquired immunity. It is a specific type of immunity. Understand a few? Then this acquired immunity has three important characters. Okay, the first character is specificity. That means our acquired immunity can specifically identify a pathogen. So this is the first character. Second character is diversity. Diversity means a difference. Our immune system, acquired immunity, can differentiate or recognize different varieties of microorganisms. Okay. Third one, distinguish between self and non-self. Already I told you. Okay, so this acquired immunity can recognize which molecule is self molecule and which molecule is foreign molecules. And this immune system, this acquired immunity giving response only to the foreign particles. It will not give any response to the molecules which are present in our body. So specificity, diversity, distinguishing between the self and non-self. These three are the important characters of this acquired immunity. Understand, students? Then, the components of acquired immunity. Acquired immunity has two important components. Okay, one is cell-mediated immunity and second one is antibody-mediated immunity. Cell-mediated immunity and antibody-mediated immunity. These two are the important components of the acquired immunity. Here, cell-mediated immunity means if a pathogenic microorganism is destroyed without producing any antibody, it is called cell-mediated immunity. Okay, so normally the antibody molecules only produced inside the body and these antibody molecules only destroying the microorganisms which are entered inside the body. But sometimes without production of antibodies, the microorganisms are destroyed. So when a pathogen is destroyed without producing any antibody, so this type of immunity is called cell-mediated immunity. And the cell-mediated immunity power is given by B cells, antigen-presenting cells and T helper cells. T helper cell, antigen-presenting cells and B cells are responsible for the T cells, natural killer cells and macrophages. Okay, T cells, natural killer cells and macrophages are giving this cell-mediated immunity. Then antibody-mediated immunity. Same here, antibody-mediated immunity is the second component of the acquired immunity. Here, when a pathogen is destroyed by producing an antibody, it is called antibody-mediated immunity. And this immunity power is given by the B cell, then T helper cell, antigen-presenting cells. Hope you understood. So cell-mediated immunity is given by T cell, natural killer cells and macrophages. Antibody-mediated immunity is given by B cell, antigen-presenting cells and T helper cells. Understand? So I hope you all understood the two components of the acquired immunity. Then types of acquired immunity. Active acquired immunity and passive acquired immunity. Active and passive, these two are the important types of acquired immunity. Okay, so here active acquired immunity means this type of immunity is produced inside the body through the production of antibody. Okay, that means when a pathogen enters inside the body, or if we put any vaccines inside the body, immediately our immune system producing the antibody. So antibody mediated, this active immunity power is produced after the entering of microorganism or after the entering of vaccines. So this immunity is produced through the production of antibody. Then passive immunity, passive acquired immunity means here our body will not produce any antibody. The antibody is injected from outside. So artificial antibodies are injected inside the body. So after that only we are getting the passive acquired immunity. Understand, my dear students? So active and passive acquired immunity are the two types of acquired immunity. Here this active acquired immunity is produced through the production of antibody. Here immunization or after the infection only it takes place. It occurs. But the passive acquired immunity is produced when an artificial antibody is injected from the outside. Understand a few? So I hope you all understood. Okay. Then now we are going to study about one important 3-mark question: the difference between active and passive immunity. Difference between active and passive immunity. See, listen very carefully. The first point: this active immunity is produced actively by your host immune system. Here host means our body. Okay, our body only the host for the microorganism. So our body playing a very important role to produce this active immunity. But the passive immunity, the host participation is not happened there, not involved there. Okay, already I told it is injected from the outside. So the first point, the active immunity is actively produced by the host immune system. Here no active participation or no active host participation in passive immunity. Then active immunity is produced due to the contact of pathogen. Already I told you, when a pathogen is enter inside, immediately active immunity is produced. But here what I said, after the injection of artificial antibody from the outside. So it's a second difference. Then the third difference between the active and passive immunity is, see, this active immunity is a durable and effective protection. That means it can give protection for more time. But the passive immunity is transient and less effective. Short period and the effect of this immunity is very less. Then the fourth difference between the active and passive immunity is same. Memory is present. When this active immunity encounter a microorganism, that instant is stored in the memory of the immune system. But here in passive immunity, no memory. Okay, no memory. That instant the encounter process is not stored in the memory. Okay, then the next one is, see, subsequent dose is very effective. Okay, once the antibody is produced, after few days or few months or few years, we have to give the second dose also, booster dose also. So when we give the subsequent second and third dose, it is very effective. This immunity is very effective. But in passive immunity, the subsequent dose is less effective. It's not give full effect. Okay, then the last point about between this active and passive immunity is, say, produced after short period. Okay, so active acquired immunity is produced within short period. Okay, after short period, but it is this passive immunity is produced immediately. Okay, because artificially the antibody is inserted. So now we have completed one important 3-mark question: the difference between active and passive immunity. Okay, and the last question: types of immune response. The immune response are classified into two important types. Okay, immune response are classified into two important types: the primary immune responses and the secondary immune response. Okay, primary immune response are produced when a pathogenic microorganism come into contact for the first time. Okay, in the first time when microorganism is coming to contact with our immune system or with our body, immediately the primary immune response is produced. Primary immune response is produced. Okay, so during this time our immune system learn to recognize the antigen, learn to recognize the foreign particle, or learn to recognize the microorganisms. and finally antibodies are produced. Finally antibodies are produced. And the second type of immune response is secondary immune response, already told you. Here, the secondary immune response is produced when same microorganism coming to contact with the body again and again. Second time, third time, fourth time, continuously. So the subsequent time, the same pathogen is coming to contact. Here one response is produced. This is called secondary immune response. This is called secondary immune response. Okay. So during this period, see, during this secondary immune response, the antibodies are immediately produced, okay, and within 2 to 3 days the antibody concentration is sharply increased. The concentration of the antibody is increased within 2 to 3 days. If you understand, so the primary immune response is produced when a microorganism is coming to contact for the first time. But here secondary immune response is produced when the same microorganism is contact in the subsequent period. Understand of you? Then now also we are going to study about one important 3-mark question. The difference between primary and secondary immune response. Difference between primary and secondary immune response. Listen very carefully. So the first points about this primary immune response is, see, this primary immune response, already I told you, it is produced when the microorganism contact with the first time, okay, but here subsequent contact, second, third, fourth time, when the same microorganism is contact or affect, immediately the secondary immune response is produced, the first one. Then the second difference between this primary immune response and the secondary immune response is, say, antibody concentration is high for 7 to 10 days. The concentration of the antibody is high 7 to 10 days, okay, but here the concentration of the antibody is high for 3 to 5 days. 3 to 5 days. So this is a second point. Then third point, say, prolonged period is needed to show its effect, to show the immunity. That means more days or more periods needed or required to show the effect of immunity. But in secondary immune response, very short period is enough. Within short period, this secondary immune response is produced. Okay. Then the next point. Here, antibody level is slowly declining. After producing this primary immune response, the antibody level is slowly decreased or declined. But in secondary immune response, the antibody level is high for longer period. Hope you understood this point. The level after showing the primary immune response, the level of the antibody is slowly reduced. But here the level of the antibody is constant, okay, is for high for long period. And the last difference between this primary immune response and secondary immune response is, this primary immune response appear in the spleen and lymph node. Spleen and lymph node. Spleen and lymph node are the example for the secondary immune organ or secondary lymph node organ, isn't it? Okay, so these primary immune responses first appear in the spleen and lymph node, but the secondary immune response first appear in bone marrow, followed by the spleen and lymph node. Followed by the spleen and lymph node. Understand, my dear students? So now we have completed two important 3-mark questions: the difference between active and passive immunity and the difference between primary and secondary immune response. Okay, and we have completed some 2-mark and 3-mark questions also. So I hope you all understood today's topic. All study well. We will meet in the next class. Thank you.